invite us down into the conference room. I know we all have to watch it.
60 seconds. Go. Okay. Welcome to the City Club of Eugene's May 13th, 2022 program, UVO Women in Science, the annual celebration of undergraduate researchers. This is the 30th program in our 21-22 programming year. My name is Kitty Piercy, and I'm the City Club President for a short time more, and very proud to be so. Support for the City Club is provided by our members and sponsors. You can become a member of the City Club at our website, cityclubofeugene.org. This program is live streamed to our YouTube channel and archived there and on our Facebook page. Our podcast is available wherever you listen to podcasts. Programs are broadcast on public radio station KLCC on Monday nights at 7 p.m. Today, we would like to congratulate KLCC on winning 5 Edward R. Murrow Awards from the, I give him a little hand here, from the Radio, Television, Digital News Association. For the 4th consecutive year, KLCC News received the award for overall excellence in the small market radio division that includes Oregon, Washington, Idaho, and Montana. The overall excellence entry included a newscast by Love Cross and reporting by Brian Bull, Rachel McDonald, Chris Lehman, Mallory Gay, Tiffany Eckert, and Aubrey Bulkley, I believe it's pronounced. Individual awards included excellence in diversity, equity, and inclusion for reporter Brian Bull's coverage of missing and murdered Indigenous women. Brian also received the award for excellence in writing. KLCC News anchor Love Cross was recognized for excellence in sound for her piece, Sounds from the Ball Game, Emerald's Return to PK Park. Cross's work also topped the competition's newscast category, which included contributions from Brian Bull, Tiffany Eckert, and Tom Vance. So let's give them a hand and appreciation. City Club has both business and in-kind sponsors. Our diamond sponsors are Kaiser Permanente, University of Oregon, Peace Health, and Lane Community College. And we get generous support from the City of Eugene and from Lane County. Our format today includes a question and answer period with City Club members and non-members alike, via live stream and text. The texting number to submit your question will appear at the bottom of your screen during the intermission and the Q&A segments. In today's forum, Women in Science, the annual celebration of undergraduate researchers, you can look up the biographies of our speakers today on cityclubofeugene.org. And now I want to thank Mary Layton for coordinating today's program. And here's Mary to introduce you to our speakers.
Do pigs have a proper role to play in filbert orchards? Is the arrival of a Whole Foods store the cause or the result of gentrification? Does a technological miracle of modern science work as miraculously in Africa as in Oregon? What kind of public health messages increase willingness to follow COVID-19 mitigation procedures? In recent years, we have heard these and other interesting questions answered in City Club programs by University of Oregon undergraduates. Participation in research is a vital part of the undergraduate experience at the university. Each year, hundreds of students from across academic disciplines can be found investigating, discovering, creating, and innovating. Research experiences allow students to explore and clarify their academic and professional goals while developing critical thinking and important workforce skills. The Undergraduate Research Opportunity Program, managed by the Office of Vice President for Research and Innovation, encourages and supports undergraduate research and creative scholarship in all the academic disciplines included in the university's 8 undergraduate schools and colleges. The program promotes inclusive excellence by working to ensure equitable access to opportunities, benefits, and resources to all students, faculty, and staff. Undergraduates engage in serious work, apprenticed to serious researchers, in studies of importance to all of us in the state and maybe on the planet. This year, 4 students, all women from the Clark Honors College, will describe what they are doing. They will remind us that college life is not just about sports and partying, though we may recall those things had a role in our own experience, but is for many, if not most, a place to invest adult intelligence, sharpened by education and mentoring, in meeting very big challenges. Be warned, you may need Google Translate. These are very smart cookies. If you are watching the live stream, questions may be texted to the number that appears at the bottom of the screen when the questioning begins. We will hear first from Sanjana Basak, a sophomore, majoring in biochemistry and a member of the Ambadi Lab at the Knight Campus for Accelerating Scientific Impact. She is a 2022 Knight Campus Undergraduate Scholars Fellow, 2022 Goldwater nominee, and 2021 First-Year Research Experience Award recipient for her research work in ophthalmic biochemistry with faculty members Dr. Bala Ambati and Dr. Hironora Uehara. Her work involves developing a targeted gene therapy for treating late-onset Fuchs endothelial corneal dystrophy, a genetic disease that results in loss of vision. Specifically, Sanjana examines how the recombinant endonuclease optodicer cleaves CUG RNA accumulation in corneal endothelial cell nuclei. Outside of the lab, Sanjana enjoys watching shows, baking, reading, and taking long walks when the sun is out. Sanjana?
Thank you very much for the kind introduction. As she said, I'm Sanjana. I'm a research assistant at the Ambadi Lab. Our lab exclusively researches eyes. Oh, thank you. Thank you. So our lab researches eyes, but I specifically study the cornea. As a review, the cornea is the outer layer that refracts light to focus vision. It's also a barrier against germs and dirt. And specifically, I researched the corneal endothelium, which is a structure within the cornea. This is a single layer of cells along a structure called the stroma that pumps excess water. Ooh, oh my gosh. All right. Sorry, some trouble with the slides. So that information was relevant because I study a disease called late-onset Fuchs dystrophy. This is an inherited disease that affects the corneal endothelial cells. When these cells die off, they damage the pumping function that pumps out excess fluid, and it leads to corneal swelling, clouding, and significant loss of vision. Don't worry, you're not in danger of this disease. As I said, it's inherited, and it only affects 4% of the US population. Nevertheless, it's a very painful disease, so that's why we're interested in studying it and treating it. So there's early onset and late onset Fuchs dystrophy. What's special about late onset is that there's an expanded trinucleotide sequence, specifically CTG, and this leads to an accumulation of CUG, cytosine, uracil, guanine, RNA in the nucleus, and this is toxic to the cell and it causes the cells to die. Furthermore, RNA splicing is disturbed when there's this RNA accumulation. So our approach to solving this RNA accumulation that's toxic to the cells was to try to modify a protein that already exists to fix this problem. There's a protein called dicer, and it is already known to cleave or cut long CUG RNA. And what we did is we modified this protein so it cannot receive negative feedback. So when the body tells it to stop cutting, it won't do that. It just continuously cuts. And our question was, can it effectively reduce long CUJ RNA accumulation and save cells from dying? Here's just a little more information about what it looks like compared to the original protein. As you can see, it's significantly smaller. This helps as we introduce it to cells. So our methods for testing Optidicer was to introduce it into cells affected by Fuchs. And then we saw the effects and how the treatment was working with a procedure called hybridization. So this is a common procedure, and it's used to detect trinucleotide repeats. So we could see if the treatment was affecting that long CUG repeat. And there's this RNA probe. This is a little specific, but when it sees that CUG RNA repeat, it will fluoresce. And if you're accessing the slides right now, they are available online if you wish to see them. You can see that the CUG RNA accumulations show up as these red fluorescents. On the photo that says control, these are the cells affected by Fuchs, and there's a lot of these red fluorescents. And then on the other picture that says Optidicer, we've introduced the treatment, and hopefully you can see that there's way less of these red fluorescents. And so we concluded that CUG accumulation was significantly reduced by this treatment of Optidicer. And we're hoping, we haven't tested this yet, that Optidicer may correct RNA missplicing and hopefully restore loss function of Fuchs cells. And this would be very valuable to the scientific world because the only way to treat Fuchs dystrophy right now is through corneal transplantation. And this is a very painful procedure, and recuperation is not always successful. And so we're hoping that we can provide a non-invasive, significantly less painful way to treat this disease. Thank you.
Next up is Carly Fear, a senior majoring in biology and minoring in chemistry. As a member of the Hosenidae lab in the Knight campus for accelerating scientific impact, Carly leads a project entitled Protein Design for Bone Healing. Funded by the University of Oregon Vice President for Research and Innovation Fellowship and the Knight Campus Undergraduate Scholars Program, Carly's research focuses on improving bone fracture healing by applying biochemical principles. Specifically, she engineers proteins with a variety of methods to control the capture and release of molecules that direct healing from customizable materials. Outside of the lab, Carly is dedicated to making scientific research accessible to her peers at the University of Oregon as the co-founder of the Scientific Research Society and a peer mentor in the Affiliated Students for Undergraduate Research Engagement. In her free time, she also loves to kayak, hike, and paint birdhouses.
Thank you. Hi. All right, let's see if I can. Thank you so much for that wonderful introduction. It's lovely to be here, and I'm super excited to talk to you all about my research. Can you all see that okay? Okay. So, as was mentioned, my name is Carly, and I am an undergraduate research in the Hosenidae lab at the Knight campus for accelerating scientific impact. And specifically, my work revolves around protein design for bone healing. So what does that mean? And I like to point out that bones typically heal relatively well on their own. So bone healing progresses through stages of inflammation, and then bone growth, and then bone remodeling, to finally result in a nicely healed bone that is able to withstand whatever kind of activities that you want to do. And so that takes usually a matter of months, and the process goes smoothly. However, in some cases, time does not heal all wounds. So for 10% of fractures, the result is called non-union. And this is a condition where the bone actually will not completely heal or bridge on its own. It needs further medical intervention. And so that's where my work steps in, trying to figure out how we can get these bones to finally heal. So zooming in a little bit on what results in that healed bone, I like to think about it as a diamond, and there are 4 sides that contribute to bone healing. These 4 sides are encapsulated by cells, matrix, proteins, and motion. So cells are those bone-forming cells. Matrix is essentially the gel-like, can you see the slides?
I think you need to hit present.
I think it might be presenting on a different screen.
And they will delete this from the tape. Don't worry about it.
Let's see.
I think you might want to yeah.
Oh, sorry. Why is it not presenting?
No, yours was working. I think it's presenting on another screen somehow.
You check your desktops.
I promoted the slides.
Oh, okay. That's what has to happen.
Yeah, cool. It's working now.
Now it's working.
Yeah.
Well, I don't know where mine is.
Yeah, it's just on the other one.
Okay, and now hit present.
Yeah, and if you hit present now, it'll work.
Okay.
Try to move back and forth in the slides just for.
Yeah. Okay, perfect. I think now it would be easier for everyone to understand what I'm talking about. So maybe we'll just flash forward to most bones heal on their own, but some do not, unfortunately. And when those bones are not likely to heal on their own, we can come in knowing that there are 4 sides that we can approach: cells, matrix, proteins, and motion. And specifically, my work looks at the proteins side of this diamond. And my favorite protein is bone morphogenetic protein 2, or BMP2. It's this wonderful butterfly-shaped protein that has osteoinductive or bone-forming properties. And so the goal of my work is to be able to deliver BMP2 to bone fractures that are unlikely to heal on their own. And the setup would look something like this. We would apply this bone morphogenetic protein, or BMP2, to a construct or a scaffold, apply this into a large fracture that is supported mechanically, and then hopefully watch healing. And in order to do this, we really need to have fine-tuned control over how quickly and where BMP2 is released. Otherwise, we might get abnormal bone growth. And so in looking at this problem, I look a lot at affinity interactions between proteins. And the way that I think about this is kind of like magnets. A magnet attracts certain types of metals through magnetic forces. And similarly, certain proteins attract other proteins through intermolecular interactions. And so these intermolecular interactions allow for us to go in and control how quickly and where BMP2 is released into this bone fracture. So a tool that I use is computational modeling in order to do this. And computational modeling is kind of a weird term, so I like to start off explaining it with a metaphor where, say you're trying to figure out what puzzle piece in a pile matches the puzzle piece that you have in your hand. Well, you could do this experimentally by picking up every single puzzle piece in the pile and trying to match it, right? You could also technically do this computationally. And it works basically like if you were to tell a computer how puzzles work and how puzzle pieces work. Instead of relying on harder experimental methods, you allow computers to use mathematical and statistical models to be able to understand the principles of the pieces that you're working with. So instead of puzzle pieces, I work a lot with proteins, and we know a lot about how proteins are made. And so computational models are super useful for figuring out how to design affinity interactions. I start off my design using all types of different protein shapes. So proteins come in all different shapes and sizes, and I like to have a diverse library of them to start with. And these are basically going to provide the overall 3D structure for the affinity interaction. But in order to get specificity to BMP2, I use structural elements called motifs. These motifs are pieces from an actual binding interaction that occurs in the body normally, and we've just borrowed a small bit of that interaction and are then able to do a process called grafting to apply it into these larger, more stable scaffolds. And the result of this is specific binders that are also stable. And this is what is going to allow us to spatiotemporally control BMP2 delivery to a fracture. So looking at a couple examples of what I've worked with, I take my motifs from native interactions of BMP2 with its binding partners in the body. And then when I do this process of motif grafting, I end up with gorgeous binders that make lots of interactions across the BMP2 interface. And these interactions are things like hydrogen bonds that rely on protons, and things like hydrophobic interactions, where basically molecules can have sticky parts that like to interact with each other. And so after generating these designs, I'm hopeful that we can apply these into real live fractures and watch healing improve. Zooming back out into kind of the significance of my work and coming back to that diamond of bone healing, I want to highlight that this has not been a solo project whatsoever. My lab, the Hosseinzadeh lab, focuses a lot on the protein end of it. But for cells and matrix, our close collaborator, Marianne Hediarachi, that is really her domain. And this project has been a huge collaboration with her in particular. And then for the motion side of the diamond, mechanical stabilization and stimulation is really a strong point of Dr. Ketgi Ong, who I've also worked with at the Knight campus. So my work has been incredibly collaborative and, of course, incredibly fun. And I'm just really happy that I was able to share what I've been able to do with all of you today. Thank you.
Thank you, Carly. Our next speaker is Audra Mcnamee, a senior majoring in math and computer science with minors in comics and cartoon studies. And for some reason, this reminds me of a saying I read, like 40 years ago about in education. Those who can teach, those who those who can do, those who understand teach. What we're going to hear next is how someone who understands uses art to teach. So one kind of transformation of knowledge that presumes understanding. And then our last speaker will talk about another kind of application. Audra's honors thesis is called Welcome to Computer Science, Designing a Comic Tour of Computers and Computing. Her research mentor is Kathleen Freeman. Outside of school, she makes short comics for fun, now listen to this, on topics ranging from the 60-year history of a libertarian billboard near I-5 to an abridged history of Jell-O. Audra, a Portland native, hopes to keep doing projects like this in the future by making educational comics for a living. Audra?
Thank you so much for that lovely introduction. I am going to mess around with my slides for a moment, and I'm going to see if the play button works. Beautiful. Okay, we're all set. As stated, I am Audra Mcnamee, and I am going to be talking about a 56-page comic that I created for my Clark Honors College in Computer and Information Science thesis. It is called Welcome to Computer Science. If you are listening to this instead of viewing it, I would appreciate your collaboration with me as we visualize some images together. But of course, you can always see the slides that I'm going to be presenting on YouTube. So, to begin, what is a comic? Is it superheroes, like in a beat-em-up? Is it a newspaper strip? I would like to propose one definition of comics from Scott Macleod in his 1993 Understanding Comics. A comic is juxtaposed pictorial and other images in deliberate sequence. I don't know if that helped at all. Kind of stepping back and turning that into words like that make sense. A comic page has multiple images on it that are in a sequence that tells a story through time. For the sake of my comics have words on them as well, and the key way that those words interact with the images is that they are on level footing. The words are as important as the images. They're inextricably linked. Without one, you get no sense from the other. Comics are just a form. They are a way to encapsulate information. You can put anything in them. You can put a superhero story. You can put education about computer science. So knowing that, why do you bother with educational comics? There are not very many studies on educational comics currently, but there are some. And the things that they indicate are that comics are a powerful way of educating because comics are narrative. This isn't inherent to comics, but many comics do choose to tell their information in a narrative form. And this helps keep readers engaged and gives them a way to process information as it builds in complexity. Comics are visual. Perhaps this set of presentations has illuminated to you the inherent visual nature of science. It's very hard to explain science clearly and concisely without images. And so having a format that is natively visual allows the images of science to shine through. Comics are reader time controlled. This is sort of a facet of visual. But of course, there are lots of visual ways of presenting information. One of them is a video. Another is a presentation with visual elements. But these are controlled by the presenter. If perhaps I'm going too slowly and you want me to hurry up, you'd have to yell at me and make me do that, and that would interrupt the entire flow of this. If I'm going too quickly and you want me to slow down, in a video you could skip back, but then everything would be repeated and you wouldn't, again, get an uninterrupted flow of information. Comics, however, you can read a comic page as slowly or quickly as you would like, and you still get the flow of information as the creator intended. Comics are also approachable. This is partially because of comics' sort of stereotypical existence in our society. They're for children, they're not very serious, maybe they're funny. Because of that, you can put educational information inside a comic, and people who might not otherwise think that they are interested in or capable of engaging with it in a textbook or a scientific article format. You can teach them the information and possibly set them on a journey of learning more. However, comics' visual nature also makes them inherently approachable. When you open them up, you're already immersed. You don't have to begin decoding chunks of text. There's other scientifically positive teaching reasons why comics are useful, dual coding and concreteness fading, but I don't need to explain that now. So, comics are good for education. Why aren't there more educational comics? There are a lot of educational comics. Every single educational comic I've read has taught me something. They're all beautiful. There are not, however, very many educational comics about computer science. There are children's picture books about computer science. There are illustrated zines about computer science. But most of the comics about computer science are either biographies of people tangentially related to the field, or they're children's comics about introductions to programming. All of these are very useful. They're excellent comics. They teach you a lot. However, there is a real gap in the available comics for a comic that introduces computer science to a high school, undergraduate, adult audience. And that is what I tried to fill. So there were a lot of directions that I could have taken this comic in. I could have done an introduction to programming. However, like this is a common way to introduce computer science. However, programming is best taught by programming, and it's very hard. It's possible, but it's very hard to program on paper with pencil. So programming classes, best left to classes, interactive web pages. The direction I chose to take instead was to introduce the breadth of computer science. So I have an introduction to the concept of computers. I have a first section, which is about computer hardware, and physically, what are the components of a computer? How does information flow through a computer? And explaining binary or electrical on and off as the basic movement of information through a computer. I have part 2, which is about programming languages, and not about how to use programming languages, but instead how programming languages are built from the simplest commands that a computer understands. Things like addition, subtraction. Computers don't natively understand anything more complicated than that, but complicated programming languages are built from these simple parts. Part 3, the final part, is about the internet, because of course, when you're engaging with a computer, it's most likely that you are in some way interacting with the World Wide Web or email or some other connective network. I both explained how information moves through the internet in simple pieces, as well as explaining functionally what the physical infrastructure of the internet is, what data centers are, where they exist in the world, how they impact the world. And that's what I did. So the comic exists. It's 56 pages. I hope, because of course, I touched on only a tiny number of topics in computer science, that in the future I can develop this into a broader project. But I am incredibly grateful to the UO Computer Science Department for enabling me to make this. And I am excited to create more educational comics in the future and learn more about what makes a good educational comic.
I'm going to read that book. Our final presenter is Jenna Travers, a junior majoring in marine biology with minors in geography, science communication, and legal studies. She works with Mark Carey in the Glacier lab, studying the effects of glacier retreat on salmon populations in the Pacific Northwest and the resulting impacts on local communities. Jenna grew up in coastal fishing towns in Alaska and Oregon, so researching the importance of salmons to communities and how they are responding to declines in populations is really close to her heart. Jenna received a Goldwater Scholarship for her thesis research, looking into how dual climate emergency like glacier retreat and salmon declines is framed for communities and stakeholders. In addition to this work, Jenna is also a writer with Glacier Hub and a salmon identifying artificial intelligence contractor. All right, I want to know what that means, actually. With the Wild Salmon Center in her free time. She has free time, I can't believe it. She enjoys hiking, swimming, and backpacking. And for fun, this summer she will spend on fieldwork, in a fieldwork position, measuring glaciers in the North Cascades, which used to take a lot longer than it probably does now.
I am going to try to get the slides to work. Help me. It might be frozen. Oh no. Oh no. Yeah, it's turning off and back on again. Oh great. Well, this part will get edited out.
Oh, there might be a mouse again.
Every so often, I think I see it,
Yeah.
and then it's not actually there.
Okay, I think we're gonna turn the computer off and back on again.
Sorry, everyone. Oh. What kind of computer is this?
It's a Mac.
That's what I thought, but it looks like slightly different.
Yeah.
I'm so sorry.
You got this, little guy. I have a somewhat comic number of windows open, so hopefully it will not take too long to return to.
Cool. That's okay.
This is why we
It's just technical
don't always
difficulties.
present things live. So,
Oh, I think it's opening all of the things.
yeah. Hi, tunnel black.
I think it's like a mark of a computer science major to have like at least like 20 pages and tabs open.
Oh, yeah. No, thank you, actually. Okay.
Okay, and then we're gonna try to present, and then
It should be up now.
do you think that's gonna show? Is it up? Oh, yay. Okay, cool. So hi, everyone. As Mary just introduced, my name is Jenna Travers, and my thesis research and what I'll be presenting on today is about stories of decline, glacier retreat, and salmon shortages. Okay, so I actually forgot that I put most of this stuff in my bio, but just a quick overview of who I am. I'm a marine biology major. I'm minoring in science communication, legal studies, and geography, and I'm planning to pursue a PhD in fisheries management after I graduate. And I'm a 2022 Goldwater recipient. My dad was in the Coast Guard, so I grew up in coastal fishing towns in Alaska and Oregon, so I've been fishing and on glaciers since I was a little kid. And so when I came to UO, I heard about the Glacier Lab, and I thought it was really interesting to be looking at glacier retreat and climate change through a societal lens rather than just a hard STEM lens. And so I joined, and my research mostly focuses on glacier retreat and salmon declines. First off, just to start, we know that glaciers are retreating in the Pacific Northwest. For those that can see the slides, there's an image of a glacier that has retreated significantly over the past 100 years in the Olympic National Park. But in addition, 35 glaciers have disappeared in the Olympics in the past 40 years. And that's not just they've shrunk, it's they have shrunk so much that they are no longer designated as glaciers. And at the same time, salmon populations in the Pacific Northwest are in a really bad spot right now. In Washington alone, 14 salmon populations are endangered or threatened, and 5 of those are deemed in crisis, to the point where if we don't do something now, they may go extinct. And so these two things are connected. Glaciers provide cold meltwater for salmon during the summer months when it's really warm, and they sustain stream flows during those times. And salmon are a cold water species, so as climate change causes their streams to warm and glaciers shrink and no longer provide that water, this is really bad news for salmon. So my research looks at some of the species-based impacts, so how will different species be impacted differently based on how long they spend in streams, their life history, life cycle, that sort of thing. I also look at societal impacts. So how will both glacier loss and salmon loss impact communities in the Pacific Northwest, either through economic reasons, cultural purposes, or ecological. And my thesis actually focuses on the third part of my research, which is science communication. And so I am really curious how these issues, and climate issues in general, are being communicated to the public. So how do we communicate issues of climate change to the public, and how does the way that we communicate them affect climate action, perceptions, all of that? And so for my thesis, I looked at the narratives and stories surrounding glacier retreat and salmon declines in the Pacific Northwest. And my principal question was, how do these change based on stakeholders? Because I thought things like, I'm going to move to the next slide, government and news organizations and environmental nonprofits and indigenous tribes would all have very different ways of framing this issue, and that could lead to some disparities in the way that these are talked about. And so I found 115 sources from 4 stakeholder groups. These sources are all publicly accessible ones, so I didn't go through the 200-page reports because those really are not aimed at the public. That's not affecting public decision. So I looked at news articles and videos produced by these and newsletters and that sort of thing, interviews. And I read through all of these sources or listened to them and transcribed them for the videos, and I took notes on narratives and themes in each of them. And I compiled these into a code book, which is essentially just a list, in this case, of the most common narratives that I found. So for those that can see the slides, those are up in the top right. But it's just things like glacier retreat will harm salmon, or glacier retreat is good for salmon in some cases. Salmon are important economically. So I just identified these common narratives, and then I used a qualitative software to go back into each one of those sources and reapply those. So I would highlight the text that showed one of the narratives, and I would just code it so that I could essentially go back later and count and see how often were these narratives stated, who stated them, why is this important. So here's what I found. It's actually not that interesting. Stakeholder did not have as big of an impact as I thought. There were some really clear trends, but I thought it was going to be this like huge difference between all of them, but they tell a similar story, and there are differences. But what I did notice is that specifically for glacier retreat and how it impacts salmon, every group except for indigenous sources mentioned at least a few times that glacier retreat would benefit salmon or that it would have mixed results for salmon. And this is important because in the far northern latitudes, glacier retreat can benefit salmon. There, the water temperatures are not rising as much because it's still cold there, and as glaciers retreat, they open up new habitat from where ice used to be for salmon. So there are these benefits. And I started thinking, why do indigenous groups, why are they the only ones that don't have these? And one of the things that I thought about was maybe it has to do with geographic location. All of the other forms have some sort of out-of-state, outside of the Pacific Northwest entity in them. So for government, there's federal government. For nonprofits, there are like international and national nonprofits. And for news outlets, we have like the New York Times and all of that. And this ended up being a really clear impact because indigenous sources, all of them came from within the state of Washington. There were no outside sources. And when I looked at geographic location, just looking at in-state versus out-of-state, I saw these really clear differences. So when talking about how glacier retreat will impact salmon, in-state sources almost never bring up potential benefits or mixed results. They are only focused on the harms because that's what glacier retreat will do for salmon. However, in future action roles, the in-state sources focus instead on the future action, whereas out-of-state sources were much more likely to talk about what was currently being done, what could be done, what strategies should be used. And so a lot of this comes down to both audience and priorities. The New York Times is not a great place to tell someone how they can get involved in this, because if someone from Tampa is reading about salmon declines in Washington, they can't go out and restore a stream. Like, there's not that same accessibility. And so the in-state source is really focused on that future action because they are the ones doing it. These state government, these indigenous groups are doing monitoring projects. They are collaborating on different climate reports. They are the ones actually doing this future action and work. And additionally, priorities are a really important thing. So for some groups from out of state, glacier retreat will benefit salmon. In Alaska and British Columbia, it can help. And so by focusing on potential benefits of it, they can know that they need to protect the habitat that will be exposed before its actual stream habitat. And so there are just these different priorities that come with that. The second main thing that I found is that overlapping climate narratives can transform narratives. And so a lot of times, climate narratives are talked about as sole things. So glaciers are retreating, salmon are declining. Those are separate. And these narratives are actually really important because narratives of glacier retreat tend to be very hopeless. Glaciers are retreating, it's gonna hurt communities, and there's very little future action. And when I looked at this, there was almost no way to help. And the reason for that is because even if we stopped emitting all carbon right now, glaciers would continue to retreat. We are past a threshold where a lot of glaciers are going to shrink and disappear by the end of the century, even if we stop immediately. And so this leads into kind of this doom and gloom narrative of like, there's nothing we can do, climate change is gonna just like ruin everything. But salmon decline narratives have a lot more actionable items because salmon can come back. We've seen after removing dams, after creating cold water refuges, that salmon populations can rebound and that they can come back. They're a very adaptable species. And so there's a lot more future action in those narratives. And when we combine the issue of glacier retreat and salmon, we can change a hopeless narrative of glacier retreat into one of action and hope. And we can change the entire narrative around climate change through this by essentially creating like, this is something bad that's happening, but it doesn't mean that it's like all these communities will suffer. And so this works for all the things that glacier retreat impacts. And it has huge implications for science communication. And that's all. Thank you.
Thank you, Sanjana, Jenna, Audra, and Carly. We'll now take a break for about 5 minutes so people can think of questions. I know I have a couple myself. And there should be on your screen, if you're live streaming, a number that you can text in. Otherwise, just write them down. Or you can just raise your hand, actually, if you're in the room. Thank you. Welcome back to City Club of Eugene. We're now ready for questions from the audience. First questioner, I see Mary.
Thank you. My first question is, well, my only question.
Identify yourself.
Oh, I'm Mary Vuxit-Schaefer, and I'm impressed with all of you tremendously. And my only question is for Jenna. Can you talk about actionable items for salmon restoration?
Yeah. So the salmon are impacted by climate change, dams, and overfishing for the most part. And so actionable items on the climate change end include restoring streams, so increasing shade and planting trees around, increasing woody debris within rivers, because that just provides more like cold water refuges for the salmon. Other things that are really helpful, removing barriers. So in Washington, there are a lot of dams, but there are also a lot of culverts, which are just small tunnels that go under the roads and are big enough for water, but not big enough for salmon. So there's a lot of projects right now that are making those larger so that salmon can actually pass through them. And so those are like kind of the main two. There's also just in general fishing management practices that we really need more collaborative fishing management projects focusing on bringing in community stakeholders and more of that government level together rather than just having it be a top-down sort of thing. Does that help? Yes.
Hi there, thank you all. My name is Karen Knudsen, City Club member and President-elect. Sanjana and Carly and Audra and Jenna, you all are excellent teachers. Just wanted to acknowledge that and really appreciated all of your presentations today. Audra, my question is for you. I found what you were sharing with us about the role that comics might play in explaining very complex information to adult audiences to be really interesting. I read years ago that some countries that have sort of the best metrics for literacy in young children, one of the things that they notice is that there's no sort of hierarchical distinction in the reading that those children are doing. It's not like a graphic novel is bad or lesser and a chapter book is better. It's really the love of the game and the engagement with the material. So I'm wondering if you could talk a little bit about why there is so much stigma attached to learning with words and pictures for people who are older. And if you see any organizations that are really innovating in this way and combining visual and written information.
I really like that question. I think that to a large extent, I don't know the history of why comics have the status that they have in like the United States today, why that all came about. I think largely it's a vestige from the panic in the early 20th century through the mid 20th century around comics, similar to how the panic about video games in the early 2000s sort of vilified them as something that was making people violent. There was this panic around comics as well, and societally we have just sort of held on to it, along with the fact that comics, to a large extent, arose in the United States as a media for children versus there's so many other comics traditions across the world that put comics in a different place in society and have comics about more different topics traditionally. Superhero comics just really were such a big force in the United States for so long that it makes sense that that's oftentimes how comics are seen here. Overall, like, we're just truly in a beautiful age for comics. There's so many organizations. There's so many traditional publishing houses, like Scholastic, like Penguin, that are putting out comics. And then there's so many specific comics presses that are putting out comics on all topics, including educational ones today. So I don't think I can indicate any one organization because like we're in this such a beautiful, bountiful moment. But really the West Coast and like sort of Portland in particular is a real hub for comics publishing in a way that's absolutely remarkable. So I think we're only going to see more amazing things coming in the realm of comics.
Thank you.
Thank you.
Greetings, I'm Paul Thompson, a City Club member for some indeterminate time. Anyway, my question relates to the scientific method. I'm a great fan of the scientific method, being a retired engineer, but sometimes it's not your friend because the discipline that it imposes and so on stretches things out in time or challenges some of your basic thoughts. So I wondered if you'd reflect for a moment on how you use the scientific method in your research and whether it was your friend or enemy.
I can sort of take a stab at this question a little bit and then I can pass it off, but I am kind of in a crossroads research discipline.
This is Carly.
Hi, I'm Carly. That is somewhere between protein engineering and classical biochemistry and molecular biology. And so my work is very collaborative, and I do have to sort of allow scientific inputs from all these different fields to influence me. And the linearity of that process can be challenged by the fact that it is so cross-disciplinary all the time, and there are so many different angles to approach from. But I think that that is, for me, one of the funnest parts. And when I'm troubleshooting experiments or designing new code, it's fascinating to just think about questions that kind of fall outside of the typical questions asked because of the nature of the problem.
Yeah, I think this is Jenna. Another thing that really came out of mine is that the scientific method is cyclical. And I think that's something really, a lot of times it's just hypothesis, experimental design, experiment analysis, and you're done. But for me, my hypothesis, my original one, was not correct. I thought stakeholders were going to be this huge impact, and I actually found things that were more important. And so it's kind of that hypothesis, experiment, analysis, go back to hypothesis. And I think that cyclical part of the scientific method, while it takes a little more time, is also really cool for kind of finding out new things, and that's how we just make these discoveries.
Hi, I'm Sanjana. I think we very strictly follow the scientific method. We have a very clear protocol that we follow. We introduce our treatment to the cells. We hybridize them to visualize if the treatment is cutting down on those CUG accumulations, and we can see those on a microscope. And after we get good results, we're not just that's it. We're currently working with 3 cell lines, and we're only halfway through working with the second one, and I've been working on this project for basically a year. And so we're all about replicating results, and even if something seems really exciting, we're always like, no, we have to try to improve these results so they're as clear as they can be.
This is Mary. I was interested in Jenna in particular because it looked to me like, Jenna, you discovered the nature of the phenomenon you were studying was different than what you expected. And so you use methods appropriate to what you think is the nature of the thing, and then you discover, wait, no, reports coming from different places matter. And so it sort of broadens out the scope.
Mm-hmm. Yeah, I think that's really important, and kind of luckily, the way my data was set up, I had a pretty good number of sources from both in-state and out-of-state, but if that hadn't been the case, I would have had to go find all new sources because my initial hypothesis was just not correct.
So I have a question, and all of you may have answers. It struck me listening to you that the impacts of your work in various disciplines affects people in different ages differently. So I'm looking around at a runner and a couple of people who are at least as old as I am, which means we're worried about breaking things. And other people are worried about their eyes for one reason or another. And I wonder if you could hypothesize, are there certain age groups that are particularly going to be affected by the work you're doing now?
Working in a place like the Knight campus that has a lot of focus on regenerative medicine, regeneration is a problem that we look more into as the population ages and as an individual ages. And so I absolutely think that age is a factor. It's difficult when you're looking at the very, very beginnings of your studies to factor in age, but later on, hopefully in, for instance, a clinical trial, that would definitely be something that I would consider.
So the disease I study, late onset Fuchs dystrophy, specifically affects people who are 50 years or older. And this is an inherited disease, so it affects whole families. And you can just think that when someone who's around 50 or older starts to feel the effects and is losing their vision, their children and their grandchildren and their younger relatives can see that that's their future, and that's probably very frightening since it affects whole generations of families.
My work is, this is Audra, and my work is based on education, and I'm really interested in pushing the bounds of who comics and who comics education are for. And so, to some extent, my work is age-based in that I am trying to make work that is interesting to people who are older than comics readers typically are, or who have grown up and not even considered reading a comic. And so I don't know how much I'm considering age in my work.
We are out of time. I'm sorry, Ruth. Okay. And welcome Kitty Piercy, president of City Club, for one more week, back to the podium to send us off.
Thanks, Mary. So this has been our May 13, 2022 program, Women in Science. Thank you, Women in Science. We enjoyed you very much. Before we, well, I just thanked you, so I don't have to say that. I'd like to recognize our diamond sponsors. Kaiser Permanente exists to provide high-quality, affordable healthcare services and improve the health of our members and the communities we serve. More information is available at www.kp.org. The University of Oregon has helped Oregonians question critically, think logically, reason effectively, communicate clearly, act creatively like them, and ever since they started in 1876. Get more information at uoregon.edu. Peace Health has been proud to serve Eugene, Lane County, and beyond as your hometown healthcare partner for more than 80 years. The Peace Health mission is to keep you and your family healthy. Learn more at peacehealth.org. Lane Community College transforms lives through learning. LCC provides comprehensive, accessible, high-quality educational opportunities that promote student success. For more information, visit lanecc.edu. We are grateful for generous support from the City of Eugene and Lane County. I would like to acknowledge our Sapphire sponsor, Bennett and Dean Real Estate, a full real estate brokerage offering a comprehensive range of services for buying or selling a Lane County residence. For more information, visit bennettdean.com. Today's sponsors are Lavas Cobb and Ferguson and Fife Dentistry. Thank you to our in-kind sponsors, KRVM 91.9 Radio, Pack Info and Simplified Computing LLC, Dotson's Photography, and a special thank you to our award-winning public radio station, KLCC, FM 89.7, for airing City Club programs on Mondays at 7. Our programs are always available on our YouTube channel, our Facebook page, and our podcast. We invite everyone, members and non-members, to participate in our programs in person, right here at First United Methodist Church, 1376 Olive, or by live stream on our YouTube page. More information on our website, cityclubofeugene.org. So next week, we will be having our annual Turtles Award. as well as learning about the election of our board and directors. This will be a very exciting program, and it would be nice to fill the room. So I invite each of you to think about coming back to Live City Club and join us next Friday. Thank you very much. And let's give a hand again to them.